Good day, viewers. My name is Mr. Kirenius Kivanu. Welcome to our first lesson for Biology Grade 10 and 11. Today's discussion will focus on enzymes as biological catalysts. The learning objectives are: define the term catalyst as a substance that increases the rate of a chemical reaction and is not changed by the reaction. Define enzymes as proteins that function as biological catalysts. Explain the effect of changes in temperature and pH on enzyme activity in terms of shape, fit, and denaturation. Describe the role of enzymes in the germination of seeds and their uses in biological washing powders and in the food industry. Investigate the uses of biological washing powders that contain enzymes. Let's get into it. For those who need a reminder, a catalyst is a substance that increases the rate of a chemical reaction without it being changed during the reaction. I am sure we can all agree that the complex working of the human body and many of the physiological processes taking place in living organisms would not be possible without catalysts. Enzymes cannot be seen with the naked eye. However, this graphic on the screen illustrates what an enzyme may look like. On the two graphics, the active sites are visible. You will notice that the active sites look different as well. Can you guess why? An enzyme is a protein that functions as biological catalyst. As I mentioned in the introduction, a catalyst is a substance that increases the rate of a reaction and is not changed by the reaction. To understand the working of enzymes, you must understand the difference between the substrate and the active site. A substrate is the substance on which an enzyme acts. If we use biological washing powder as an example, a protein stain such as wine or blood or egg would be the substrate that the enzyme must act on. An active site is a region on an enzyme that binds to a protein or other substances during a reaction to form an enzyme substrate complex. This graphic clearly explains how an enzyme functions. When an enzyme binds to its substrate, it forms an enzyme substrate complex. The part of the enzyme where the substrate binds is called the active site, since that it is where the catalytic action happens. The active site of an enzyme creates an ideal environment for the reaction to occur. After an enzyme has finished catalyzing a reaction, it releases its products. The enzyme will always return to its original state once the reaction is completed. Now that you have learned what enzymes and catalysts are, let us look at two factors that influence the activity of enzymes, namely temperature and pH. Each enzyme has an optimum temperature, which is the temperature at which the enzyme works best. In humans, this temperature is generally 37 degrees Celsius. When temperature increases, molecules move faster, so the rate of collision increases. Thus, the reaction can happen in a much shorter time. Having more energy makes them more likely to bind to an active site. If the temperature continues to rise or becomes too high, enzyme molecules vibrate too much and enzymes denature. The active site on the enzyme loses its shape and will no longer bind to the substrate. When the temperature is lower, molecules move slower, slowing down the rate of collision, which in turn slows down the reaction of the enzymes. Each enzyme works best at their optimum pH, as can be seen on the graph. Pepsin works best at pH 2, amylase works at pH 7, and trypsin works at pH 10. If the pH is changed, then the enzyme will denature and the substrate will no longer fit into the active site, and as a result, no reaction will occur. The next part of our discussion will focus on enzymes and their uses. Enzymes help seeds to germinate. Seeds contain enzymes, an embryo plant and stored nutrients such as protein, starch, and lipids, which are insoluble. The embryonic plant stored nutrients unless they are changed into soluble molecules by enzymes. As you can see in the graphic, the seed has germinated. The radicle is visible in the graphic because the soluble nutrients, which are the products of enzyme activity in the seed, were used for growth. In the presence of water, these enzymes digest as follows. Amylase digests starch into maltose. Maltase digests maltose into glucose. Protease digests protein into polypeptides and amino acids. And lipase digests lipids, which are fats, into fatty acids and glycerol. Did you notice any similarity in the words amylase, maltase, protease, and lipase? They all end with the letters ACE. All enzymes end with ACE. Therefore, it is always easy to distinguish an enzyme. I briefly spoke about biological washing powder earlier, so let us look at it in detail. Use of enzymes in biological washing powder. Biological washing powders contain a range of hydrolyzing enzymes. The common enzymes found in such washing powders are amylases, which breaks down starch stains, cellulases, which breaks down ends of damaged cotton fibers and remove fuzz produced during washing, lipase, which breaks down lipid stains into fatty acids and glycerol, proteases, which breaks down protein stains in food stains. The enzymes in biological washing powders help to remove biological stains, such as blood, wine, and egg stains, thus making clothes and objects clean. Now that you have come to the end of today's lesson, let us look into what we learned about enzymes as biological catalysts. Today we learned that enzymes are proteins that function as biological catalysts. A substrate is the substance on which an enzyme acts. An active site is a region on an enzyme that binds to a protein or other substance during a reaction to form an enzyme-substrate complex. You also learned that enzymes have an optimum temperature and pH where their rate of reaction is at its best. Furthermore, you learned that enzymes help seeds germinate by digesting insoluble nutrients into soluble nutrients to be used by the embryo plant for growth. Lastly, you learned that biological washing powders contain enzymes that help to clean clothes by removing stains. Thank you very much for watching. I hope you learned something new and you enjoyed today's lesson. Until we meet again, see you.